Hello students, today in this module I am going to discuss the experiment number 2 of biology that you have to do in your practical file class 10th. Okay, so the aim of the experiment is, in this experiment we have to study the binary fission in amoeba and budding in yeast. And we have to study these two things, that is budding and binary fission, with the help of the permanent slides. So we will be having the permanent slides, already prepared slides of binary fission and budding in yeast and in hydra and binary fission in amoeba, and we have to watch them under the microscope. We have to observe the things which are present in the permanent slide, and we have to write down the observations. So let's begin with the material required. So the apparatus that we require for this complete experiment is a compound microscope. Then we read the prepared slide, that is the permanent prepared slides of the binary fission, budding in yeast and budding in hydra. So only two apparatus we require. One is the compound microscope and the other is the already prepared permanent slides of the binary fission and budding. Now let us discuss some theoretical part. We already know that reproduction is the production of the offsprings, and it is of two types, sexual and asexual. We have already done in the chapter, that is how do organisms reproduce. And we have already discussed that asexual reproduction is a type of reproduction in which a single parent is involved. And there are many types of asexual reproduction, and many organisms follow different processes to do the asexual reproduction. Now budding and binary fission, they both are the methods of the asexual reproduction. The binary fission is especially in protozoans and bacteria, and budding is in yeast and hydra. So these are the common examples that do these type of the reproductions. Now under the procedure, what we will do, we will select the prepared slides of amoeba for binary fission and budding for yeast and hydra for budding. Then we will set up a light microscope, look through eyepiece and adjust the mirror and diaphragm. So very first thing that we need to do before starting the experiment is we should have the slides that we require, and the second thing is we need to focus the light on the compound microscope. So by adjusting the mirror and by looking through the eyepiece, we will adjust the light and we will make it quite clear. Our view should be clear. Then we will keep our slides on the stage, and then we will start observing the permanent slides. First we will observe the slides under low power, that is around 10x, and then we are going to watch the same slide under some high power, okay, that is around 40x. So after observing the slides, we will be able to see the amoeba, the nucleus of the amoeba and the binary fission in amoeba, that the cells are dividing. Similarly, in case of the budding, we will be able to see the tiny buds, and we will be able to see that the parent organism is attached with a tiny bud which will detach in future. Okay, so such kind of observations we will get. Now let us look at the observations more in detail. So in case of the binary fission, we will get this much observations, like it is a type of asexual reproduction in which two daughter cells are formed from a single parent. As I told you, in asexual reproduction a single parent is involved. So by looking at the slides, we will be able to observe this properly, that yes, there are two daughter cells or many daughter cells which may be formed by many amoebas which may be present on the slide, and a single amoeba give rise to two daughter cells. Now the parent cell along with the nucleus belong elongated. So we may get this kind of the view in the slide as well, that there is an amoeba in which the developing nucleus is little bit elongated. This elongated nucleus actually tells us that yes, the cells is going to divide. Okay, because whenever the binary fission take place, the very first thing is that the amoeba's nucleus, it start elongating. Now the nucleus divide mitotically first and then the cytoplasm divide. After this we can see, as look at the diagram, the nucleus elongate and then it divide, the nucleus divide, and see the nucleus have divided. After the nuclear division the cytoplasm also divide, and we can see here the two cells which are just joined silently, and after these they will divide into two daughter cells. So the nucleus will divide first and then the cytoplasm. At the point of fission, constrictions appear which deepened to divide the cell into two cells. So basically what happen is the nucleus divide, the cytoplasm divide, and there are some point of fission which are in the form of the constrictions, and we are able to see that. And these fissions lead to the complete division of the cell into two daughter cells. So these observations we will get from observing the permanent slide and the prepared slide of the binary fission in amoeba. Now let us look at what we will observe when we look at the permanent slide of the yeast. It is again a type of the asexual reproduction in which a small protuberance or outgrowth arises from the parent body called bud. So the parent body is quite mature, is quite large, and from the parent body a small bud arises, okay, like this as shown in the diagram. Okay, now this bud will, this bud will mature and it will elongate and it will increase in the size. The nucleus of the parent yeast cell divide into two parts. One part of the nucleus passes to the bud. So basically here what is going on, that the parent cell, that is the yeast cell, will divide into, nucleus of that cell will divide into two parts, and one of the nucleus will go to that bud and the other one remain in the parent cell. Okay. Now as the bud mature, it will detach from the parent body and it will grow independently as a new individual. The body may remain attached to the parent body, forming chain of cells. So it can happen that the bud may detach prior, but it may happen that after getting a nucleus and after being mature also, the bud do not detach. It remain joined with the parent body, and these kind of chains of buds are formed. So both the cases are possible. So I hope that you are clear that in case of the binary fission that we have discussed earlier, the parent cell divide into two cell, means the parent identity is lost. But in case of the budding in yeast, the parent cell always remain there, so the identity of the parent cell is not lost. Now let us discuss budding in the hydra. We know that hydra is a multicellular organism and it reproduces asexually by budding. In hydra, a bulb-like projections appear on the body, and this projection or outgrowth is known as the bud. Similarly, as in the yeast, a bud arises, in the same manner in the hydra also, a tiny bud arises. This bud grow, elongate and mature. So this bud grow in size, develop into a new individual. The daughter hydra detach itself from the parent body after completing the development and lead its individual life. So here the small tiny bud, it's keep on growing, it keep on maturing, taking nutrition from the parent body, and when it attain a good size and it get mature, it detach from the parent body and behave like a separate new individual. So in the budding in hydra as well, the parent identity is not lost because parent is still there and the new individual is formed from the parent. So what is the result of this complete experiment by observing both three type of the slides? What we conclude is that the slides show binary fission in amoeba and budding in yeast and budding in hydra. So when you will be given the permanent slides of such type, you have to observe the slide and you have to tell what you see in the slides. Okay, first in the low power and then in the high power. So when I'll give you the slide of the amoeba, then you have to observe that yes, it is amoeba with the irregular shape, and you have to observe yes, it is dividing by binary fission. Similarly, from the slides of the hydra and yeast, you have to observe what you see. If you see the yeast, it will be circular in shape and it will show some tiny bud on it. Okay, and if you see the hydra, you will see the tentacles of the hydra, and you can be able to see the part attached to the parent body, or tiny hydras detaching from the parent body. Okay, so such observations you will see. Okay, you will observe these things. Now in the overall experiment, the precautions that we need to take while doing this experiment: always set the microscope mirror and the diaphragm before placing the slide. As I told you, the very first thing while we observe a permanent slide is we have to first manage our compound microscope. So we have to keep our compound microscope in appropriate light. We have to set the mirror, and we have to see from the eyepiece, and we have to check whether the appropriate light is coming or not. We have to set the light first. Now always focus the slide first at the low power and then at the high power. So we will see the slide first at low power 10x and then at the high power, that is 40x. While focusing at the high power, do not use coarse adjustment of the microscope, use only fine adjustments. I have already discussed about the parts of the microscope with you. The microscope is having two adjustments, the coarse adjustment and the fine adjustment. So when we are doing at low power, we use the coarse adjustment, okay, but when we are using the high power, that is 40x, we don't use much the coarse adjustment. We take advantage of the fine adjustment because we just want to focus the things. We know that the thing is already there in the view. Now observe the slide, and then you have to draw the diagram. First you will observe the slide, you will see what you look, and you will write your observations, and side by side you will draw the diagram after observing the slides. So I hope the experiment is clear to you all, and this was the second experiment, and you have to write this experiment in your practical files. Thank you.